I'm going to be talking about conservation genetics of the Texas trailing phlox. Okay, so to start off with though, first I'm going to give an overview of conservation genetics and the conservation genetics program that I run at the Missouri Botanical Garden. And then next I'll show you an example how genetic data can assist in conservation using Texas trailing phlox as an example. Okay, so um, here is an infographic that shows the current biomass of Earth's land mammals. And so here in blue in the center, we have humans and our biomass. In light blue, there's the biomass of our pets and livestock. And then in green is actually the biomass of wild animals. And I think this infographic does a really good job of showing how humans and our livestock and our pets have really pushed wild species to kind of the fringes and fragments of ecosystems that they previously occupied. And it shows how humans have really taken over our planet. And because of that, we're experiencing a biodiversity crisis. So this is a study that was published in 2015, looking at the background extinction rate that happened before the Industrial Revolution, as well as the current extinction rate from the 1700 till now. And the paper concluded that the current species extinction rate is between 100 and 1,000 times the natural background rate. And they attributed it to one cause, which is competition with us. So we do things like destroy natural ecosystems, unsustainably use species, and there's also climate change. So this is actually something that is happening in plants as well as animals. So the current plant species extinction rate is around 500 times the background rate. And because of that, around 42% of plant species are threatened with extinction. And this map here just shows the kind of concentration of extinctions. And although a lot of concentrations are concentrated on oceanic islands, there's actually quite a few that are occurring kind of in the central and southeastern US. Okay, so those previous studies were looking at species diversity, but as you probably are aware, scientists recognize three fundamental levels of biodiversity, which include ecosystem diversity, species diversity, which I think is the way that people predominantly think of diversity, and then there's also genetic diversity, which is essential for the physical and functional differences among individuals in a population. And it's important because it's essentially responsible for promoting resiliency in species. So if you have a species that's genetically diverse and a disease goes through, there's a higher likelihood that there will be individuals there to withstand that disease than a population that has low genetic diversity. So genetic diversity is important. And I also want to mention that it's important to conserve all of the levels of biodiversity because they're interconnected. So, for example, if you get declines in endangered species and a loss of genetic diversity, that can cause the loss of a species. The loss of a species can destabilize an ecosystem if it's a keystone species. And then the destabilization of an ecosystem can further feedback and cause losses of genetic diversity in many different species. So it's important that we're conserving all of these different levels of biodiversity. Um, but I would argue that our knowledge of biodiversity is incomplete. So at the species level, there are lots of species out there that are taxonomically questionable, and we don't know whether or not we should be recognizing it. There's also species out there that are cryptic, and we actually don't know that they exist, but they're unique evolutionary entities. And then at the genetic diversity level, this is a level of biodiversity that's very poorly known because it's not readily observable. So we can't go out into the wild and look at the population and know its genetic diversity. Um, and the other thing is it's very difficult to predict because it's affected by things that we don't necessarily know happened. For example, things in its demographic history can affect genetic diversity. So, for example, a species may experience a big population crash and cause a a genetic bottleneck and we wouldn't know it and could have very low genetic diversity. This is like something that happened in cheetahs, for example. And then for plants in particular, life history traits can affect genetic diversity. And a lot of times we don't know that much about plant life history traits and how they affect genetic diversity. Um, However, I would argue that effective conservation requires that we have an accurate understanding of biodiversity because we can't conserve something if we don't know how much we have or what we have. And so that's where I feel like the field of conservation genetics comes in. It's essentially the field where we use genetic data to quantify both genetic and species diversity, and we also ensure that that biodiversity is effectively conserved. Okay, so here is a flyer that my lab manager developed a few years ago showing some of the projects that we have. And I just want to point out that we work on all kinds of different things. You know, we work on Texas Trailing Phlox. We work on actually another species we work on in Texas is geocarpon. You know, we also work on large trees, we work on agaves, we work on all kinds of different species. And kind of the main unifying theme across all of the different species we work on is, in general, they are mostly threatened with extinction. Um, so for plant species that occur within the United States, we work on species that are often listed under the U.S. Endangered Species Act as either endangered, meaning they're on the brink of extinction, or threatened, meaning they're likely to be on the brink of extinction soon. For species internationally, we work on things that are often listed by the IUCN Red List and categorized as things like critically endangered, endangered, or vulnerable. Um, then, in general, a lot of the projects that we work on are actually kind of sparked by collaborations with partners. So, a lot of times we work with people in specific land management agencies like U.S. Fish and Wildlife Service, Texas Parks and Wildlife Department, um, and they have specific questions they want answered. And then what we'll do is we'll write a proposal together to try to answer that question to get funding. And the types of projects that we work on are things like understanding the basic biology of plants. So, for example, what are the life history characteristics of an endangered species? So, you know, plants have all kinds of different mating systems and dispersal agents, and so genetics can provide some insight into that. A second question is, have populations experienced genetic declines? So do we see that some populations may be affected by genetic diversity? Another question is, what is the best strategy to protect genetic diversity when it's structured in different ways across the landscape? Another question is whether a species is unique and in need of protection, or whether it's actually just a subpopulation of a more widespread species and it really doesn't need protection. Um, and then another one is how much wild genetic diversity is conserved in ex situ collections, so off-site collections, not in its wild population. And then finally, another question is how do we ensure that reintroduced populations have high genetic diversity? So making sure we're selecting populations that have, or source material that is genetically diverse. Okay, so how do we collect genetic samples? So basically what we do for plants is we will go out into the wild and we will collect leaves. And generally this is non-invasive, so we'll just collect a few leaves off of a plant. Doesn't usually harm the plant. And because we're population geneticists, we generally will collect from multiple individuals in multiple populations, usually spanning the entire geographic range of the species. And sometimes a lot of times we focus on wild populations, so in situ genetic diversity, but we'll also focus on living collections in botanical gardens or in seed banks and things like that, so ex situ collections. So once we have those samples, then we bring them back to our lab. This is what my lab looks like. Um, and then we'll take the sample and we'll grind it up and we will extract DNA out of it. Then we'll take those DNA samples from each individual and we'll genotype the same DNA region from different individuals and then compare them. So then once we have that genetic data, what we'll do is we'll analyze the patterns of similarities and differences in the genetic data. So essentially we're comparing the DNA regions to each other. And then we can look at things like the genetic diversity in the population, So how much variation is there in their genetic regions? We can look at how genetic variation is structured. Um, and then we can also look at evolutionary history. Okay, so next, I will give an example of how genetic data can assist in conservation using Texas trailing phlox as an example. Okay, so this is longleaf pine savanna, and you guys are probably acquainted with it, but it's an open fire maintained ecosystem. Disturbance is really important in this ecosystem. It has an open canopy of longleaf pine, and it has a grass-dominated understory. And in that understory, there's relatively high forb diversity, and there's quite a few species that are endemic to that particular understory. How and this is a once was a very widespread ecosystem, so it once occurred all across the southeastern coastal plain, all the way from Virginia down through Florida and all the way through eastern Texas. But unfortunately, over the last century, this ecosystem has declined dramatically. So really only a fraction of the previous land is currently occupied by longleaf pine savanna. And today I'll be talking about longleaf pine savanna in Texas, where the ecosystem has declined dramatically because of several different reasons. So one is harvesting of the longleaf pine trees. The second is pine plantations of species including loblolly pines and slash pines. And then another thing is this is a disturbance maintained ecosystem, and fire suppression and lack of management can really cause declines. So you'll get kind of encroachment by a woody understory of yaupon in Texas, and that will choke out all of the understory forbs and grasses. Um, so because of the declines in this ecosystem, species that occur in longleaf pine savanna have also experienced declines. And one of those is Texas trailing phlox or Phlox nivalis subspecies texensis. So this is a suffrutescent perennial, which means that it has kind of parts that are woody and parts that are herbaceous. And it forms this kind of mat-like creeping structure across the ground in these longleaf pine savannas. So it's endemic to three counties, which are Polk, Tyler, and Hardin counties in Texas. And it's only found in longleaf pine savanna understories. And actually, when it was described as being abundant in the pine lands off of US Highway 69 between Woodville and Warren and Tyler County. So, an analysis by the US Fish and Wildlife Service found that it was historically occurred in six large sites of six very large populations based on kind of historical occurrence data. But now it's currently known from only four sites, and several of those populations are extremely small. So it's really declined over the past 50 to 100 years. And because of that decline, it was listed as endangered in 1991. Okay, so Phlox nivalis subspecies texensis is one of three subspecies of Phlox nivalis. So as I said, the first one is subspecies texensis, which is found in the yellow portion of Texas that you see there in those three counties. And it's morphologically distinct from the other two varieties in that it has very distinctly divided corolla lobes. So the picture on the left there of the flower, you can see that the corolla lobes are quite separate in comparison to the other subspecies that you see on the right that have corolla lobes that are not nearly as divided. The second subspecies is Phlox nivalis subspecies nivalis. That's found along the Atlantic Gulf and coastal plains, generally around Florida and Alabama. And these are smaller plants. They have shorter and more narrow leaves and finer gland-tipped pubescence. And then finally, there's Phlox nivalis subspecies hensii, which occurs in the Piedmont region, kind of the more inland regions like in the Carolinas. And it has more coarsely glandular leaves, has longer and wider leaves as well. So there's three subspecies and they're all morphologically distinct, but the actual recognition of these subspecies varies according to the author. So there's some authors that recognize all three subspecies. There's some that recognize two, which would be texensis, and then the other two as a single entity. And then the most recent treatment in Flora North America actually recognized no subspecies. So that kind of led to the question of whether Texensis is genetically distinct or not. And this is important because it can determine whether it may merit protection under the Endangered Species Act. So that was one of the questions that we wanted to answer as part of this project. Another question that we wanted to answer was how well Phlox nivalis subspecies texensis was meeting its recovery goal. So recently, the U.S. Fish and Wildlife Service updated their recovery criteria for this species, and essentially what they decided would be when it would be eligible for delisting is when there has 15 populations that contain greater than 16 individuals that are maintained and established. Um, so that's actually a pretty ambitious goal. And there are supposed to be at least 4 populations in each county that the species occurs in. But there's some questions that could be answered with genetic data relevant to this. So the first is how many genetically distinct populations exist in this subspecies, as well as how many genetically distinct individuals occur in each of those populations. And does the species exhibit clonality? So for example, do we have, maybe fewer individuals than we expected? Um, okay, and then finally, there's been quite a few ex situ conservation efforts for the species. So, for example, people at Mercer Botanical Garden developed a ex situ collection from material collected in the 1990s. And then that material was used to conduct essentially 3 reintroductions or reintroductions in 3 locations. But there's some questions about that too, which are, how are these reintroductions related to the wild individuals? And how much of the wild genetic diversity is conserved in these ex situ populations? Do we have all the genetic diversity conserved, or is it kind of at risk of loss? Okay, so to answer these questions, we developed a genetic study. And the goals of the study were first to assess the genetic distinctiveness of Phlox nivalis subspecies texensis relative to the 2 related subspecies, Phlox nivalis subspecies nivalis and Phlox nivalis subspecies hensii. The second goal was to understand the ploidy, clonality, genetic diversity, and structure in wild populations of Phlox nivalis subspecies texensis. So whether it's polyploid, whether it reproduces vegetatively, and then also how much genetic diversity there is and how it's structured across the wild populations. And then next, we also wanted to know how well the reintroductions conserve the genetic diversity of the wild populations. And then finally, we wanted to make conservation recommendations based on our results. Okay, so in terms of methods, I'm just going to go over general methods first and then talk more in depth about sampling. But basically what we did is we sampled populations of all of the 3 subspecies of Phlox nivalis as well as an outgroup. And we did that to look at both genetic distinctiveness, but also to be able to compare levels of genetic diversity across the rare subspecies texensis, and then the other 2 subspecies. We also we then genotyped those samples at 18 microsatellites. We estimated the ploidy, so whether it's a diploid or a polyploid, based on the number of alleles per locus at the microsatellites. We estimated clonality and genetic diversity. And then we looked at genetic structure across the landscape in 2 data sets. So one that contained the wild Phlox nivalis subspecies texensis and its outgroups, and then one that contained Phlox nivalis subspecies texensis and the reintroductions to see how they're related to each other. Okay, so this is just a little bit more in depth about our sampling. Okay, so first, I'm just going to talk about the outgroups that we collected. So we sampled 1 population of Phlox oklahomensis from Kansas. And then we sampled 2 populations of Phlox nivalis, subspecies nivalis in Florida. We sampled 3 populations of Phlox nivalis subspecies hensii in South Carolina and Georgia. And for each of these, we tried to collect at least 24 individuals per population. Okay, then we were sampling Phlox nivalis subspecies texensis. And for this, we really tried to do a really comprehensive survey of the historical occurrences of the subspecies. So we tried to visit every site that the species had been recorded at, um, and and within, as long as we had permission. And then when we found the plant, we tried to sample every distinct individual. So what we did is we would look to see if there was any sort of physical connection between two plants that might be next to each other to see if they were connected. And then if they were, we would only collect one. But if we could find no physical connection, we would collect them both. So let's see what we ended up with. Okay, so basically in one site, we found a single plant. This is a privately owned site. In the next site, which is part of a big thicket, we found a population that included 9 plants. And then we had 2 wild populations, one that contained 67 plants, and another one that contained 183 plants. And these were really closely adjacent to one another, but we considered them 2 different sites because they were separated by greater than 1 mile. So overall, we only were able to come up with 260 individual plants in 4 sites, with the sites defined as being separated by greater than 1 mile. So overall, there's only a few individuals of the species remaining, sadly. We also sampled 67 plants from 3 different reintroduction sites. One of these sites has additional individuals, but we were just trying to get a sample of the genetic diversity of these reintroductions. And in total, we were able to sample 456 individuals. Okay, one of the things I wanted to point out before I got into the genetic results is that there's always been a question about kind of how continuous the distribution of Phlox nivalis is. So historically, there have been populations that occurred in Mississippi and Alabama. And we actually went to visit a lot of those sites, and we were unable to find any populations that occurred in between Phlox nivalis in Texas and then Phlox nivalis in the Panhandle of Florida. And this is actually a map of iNaturalist observations. And generally, it looks like the species generally, the disjunction has widened. So there really is probably no populations occurring between Texas and either Alabama or the Panhandle of Florida. So overall, this species really does have a very disjunct distribution with Phlox nivalis subspecies texensis divided by a long distance from the rest of the range. Okay, so is this subspecies distinct from the remainder? So this is a genetic distance tree. So each one of these little labels here is a population, and the branches represent genetic distance, so it shows how similar populations are to each other. And so what we found is that, so here's Phlox oklahomensis at the bottom. And then Phlox nivalis separates into 3 groups, and actually the groups segregate according to subspecies. So we find a group that contains the 3 populations of Phlox nivalis subspecies hensii in South Carolina and Georgia. The second group contains Phlox nivalis subspecies nivalis, the 2 populations in Florida. And then the remaining group contains all of Phlox nivalis subspecies texensis, and then kind of further subdivides them according to whether they are wild or reintroduced. But I think the take-home message is that these results suggest that Phlox nivalis subspecies texensis is genetically distinct from the other 2 subspecies. So let's look at this in a little bit more detail. So we analyzed the data using a program called Structure. And what that does is it takes individuals and it essentially tries to place them into different groups according to their kind of genetic makeup. So, and it produces a plot that looks like the thing that you see at the bottom. So in that, basically, so each one of those lines, those vertical lines that you see, represents an individual. And the colors represent genetic groups, and it essentially shows the genetic group to which individuals are assigned. So if all of the individuals that are assigned to one color are form a genetic group. So what we can see here is, according to this diagram, we see a unique group that's made up by Phlox oklahomensis, which is unsurprising because it's a different species. But then we see that Phlox nivalis subspecies nivalis forms a unique group. And then the three populations of Phlox nivalis subspecies hensii form a unique group. And these two groups from hensii and nivalis are genetically distinct from subspecies texensis. So within subspecies texensis, we see that the individuals are grouped into three genetic groups. So that's the kind of burgundy color, and then there's the light pink and the green. So in the inset in the map, you can see where these individuals are located within the range of Phlox nivalis subspecies texensis. So what we see is that the burgundy cluster is only found in the northwest portion of the range of Phlox nivalis subspecies texensis. And these are actually those two small populations. So one of those contains only a single individual, and then another one contains 9 individuals. And then the remainder of the individuals in the subspecies are broken down into the pink and green groups. And those mostly occur in two relatively contiguous populations that are directly adjacent to each other. And so what we see is that the northern population contains mostly the pink genetic group, and the southern population contains mostly the green genetic group, but there's a lot of admixture between those, which is kind of essentially a pattern that you would expect based on isolation by distance, where, the more northern ones are pink, the more southern ones are green, and then there's a mixture kind of where they overlap. So overall, in summary, we see that Phlox nivalis subspecies texensis is genetically distinct, and it's made up of the 4 populations are made up of 3 genetic groups. Okay. So let's look at the ploidy. So we wanted to know if Phlox nivalis was polyploid. And this was based on a previous study by Zale et al., who sampled a few individuals and found that there was both diploids and polyploids in Phlox nivalis. And so what we did is we looked at the number of alleles per locus at microsatellites and used that to infer the ploidy of the individuals. And so what we found is that overall, almost all of the population showed variation in ploidy. And it ranged from things that are diploids of 2x all the way to things that are potentially pentaploid. And we also saw some variation within populations in terms of ploidy. So for example, one of the largest population of Phlox nivalis texensis, we found individuals that looked to be diploids, triploids, tetraploids, and hexaploids. So there's significant variation in ploidy. And we think that this is the case because we think it's an autopolyploid, so it's essentially spontaneously doubling its chromosome numbers. And this can be an issue because sometimes individuals of different ploidies may not be able to reproduce, so it could be potentially causing reproductive problems. But that needs to be investigated further. Another thing that I wanted to point out is that we were also interested in clonality and whether the species exhibited any evidence for clonality. And so I wanted to point out the fact that we sampled 260 individuals, but when we actually looked at their genetic diversity, we found that a handful of individuals were genetically identical to each other. So usually we would find that neighboring individuals were genetically related, and they were actually clones. So essentially, instead of having 260 individuals, we were able to actually only sample 224 genetically distinct individuals. And we think this is happening. There's 2 ways. I think either a plant is kind of maybe a little fragment breaks off and then roots next to a different individual, so that you have 2 genetically identical individuals right next to each other. Or they could actually be connected underground, but we can't tell. So they just look like they're not unconnected, but they actually may be physically connected. But in general, it does look like there is some clonality, and unfortunately, it means that there's fewer genetically distinct individuals than we thought. Okay, so next, let's look at genetic diversity. So what we're doing here is this is allelic diversity, so the average number of alleles per locus across all the different genetic loci. And what we wanted to do is we wanted to compare allelic diversity between the populations of Texensis, which are the four on the left here, with those from more widespread congeners or conspecifics. Um, and so what we found in general is that if you look at the two largest populations of Phlox nivalis subspecies texensis, which are the wild C and wild D, um, it looks like they have generally comparable levels of genetic diversity to the more widespread species, or the ones that have the highest genetic diversity. However, there's two populations. The two smallest populations have much lower genetic diversity. So one of them has an average of 1.7 alleles per locus, and the other one has 3.6. Um, Generally, we think this is indicative of a decline in genetic diversity in these populations. So it's likely that these populations have undergone genetic drift, potentially maybe bottlenecks that have caused reductions in genetic diversity. So unfortunately, we are seeing some signs that are indicative of genetic decline in these populations. Okay, so there's also some other metrics of genetic diversity that we looked at. So for example, we looked at heterozygosity, which is essentially, so observed heterozygosity, which is HO, is the proportion of heterozygous individuals that are present in the population. And then we can compare that to the expected heterozygosity, which is essentially taking the gene frequencies of each of the alleles and then putting it through the Hardy-Weinberg equilibrium equation, and then using that to get at what you would expect. So when we look at this and we compare them, we get the GIS. And generally, what the GIS is indicative of is whether inbreeding has happened. So when you have observed heterozygosity that's lower than expected heterozygosity, that suggests that inbreeding is happening. So individuals are becoming more homozygous because they're getting the same alleles from their parents. And so we get a positive inbreeding coefficient. So the GIS, the inbreeding coefficient, um, um, the GIS, the inbreeding coefficient, can range from +1 to -1. So a high positive number indicates inbreeding. Numbers around 0 indicate random mating. And then numbers that are negative generally indicate assortative mating. And what we can see is actually across all of these populations, we see that almost all populations have a positive GIS value, indicating that inbreeding is happening across all of these different populations and all of these different species. So it does look like inbreeding is kind of just a feature that is occurring across all of these different species and populations. Another thing I want to point out, though, is that I mentioned previously that there's been genetic declines in these small populations. And here again, we see that the smaller populations have lower genetic diversity. We think that these have experienced declines in genetic diversity, unfortunately, because of their small population size. Okay. So the next question was related to the genetic representation and reintroductions. And so once again, this is a structure diagram. And in this analysis, we included the wild Phlox nivalis subspecies texensis, and we also included the reintroductions. And so basically what we found, the patterns of genetic structure in the wild populations look the same. We see 3 genetic clusters, and in general, they're structured like we saw before, where 2 sites are in the maroon cluster, and then the other 2 sites are equally split between the pink and green genetic clusters. But interestingly, what we found is that the reintroductions group separately from the wild Phlox nivalis subspecies texensis. And when we looked at the actual genetic diversity in these reintroductions, what we found is they actually contained quite a few unique alleles. And we think that the reason why this is occurring is because the genetic material for these reintroductions was sampled in the 1990s, and we think that that some of that genetic diversity may have been lost in the wild population since it was collected. So actually, these reintroductions are actually conserving some important genetic diversity that's been lost in the wild. So they actually are very important. Um, and we see that the same thing at the bottom. So this is our genetic distance tree. And what we see is once again that the the reintroductions are clustering separately from the wild populations. They're kind of forming a group that's close to the source populations, which are wild populations C and D, but they are genetically unique. So another question that we had was how much diversity in the wild population is contained in the reintroductions. And so I just want to go back to the allelic diversity that I showed before. So on the left, we see the four wild populations of Phlox nivalis subspecies texensis. And then on the right, we have the reintroduced populations. And what we can see is the allelic diversity in the reintroduced populations is much lower than in the wild populations. And so what this is indicative of is that there's diversity in the wild populations that are not currently conserved in the reintroductions. So what we need to do here is we probably need to go back out, take cuttings of wild individuals and use them to augment those reintroduced populations so that they may encompass more of the genetic diversity present in the wild populations. Okay, so in conclusion, so the first question we had was whether Phlox nivalis subspecies texensis is genetically distinct. And in general, what we found is that it indeed is genetically distinct. It does form unique genetic clusters. We also found that it has a strong allopatric distribution, so it doesn't overlap with the other subspecies. And it's also morphologically distinct. And so we think that there's actually pretty strong evidence that it could represent a new species. We're still kind of working out the relationship between it and the other two taxa, including subspecies hensii and subspecies nivalis, and particularly the relationship between those other two subspecies to decide whether we should describe it as a new species. But overall, I think the kind of take-home message is it is definitely distinct, and it should be recognized. And because of that, it does merit protection under the U.S. Endangered Species Act. So it is a unique taxon, and it should be protected. The second question was related to ploidy. So we think Phlox nivalis is a mixed cytotype autopolyploid. So what that means is that we have individuals that could be diploids, triploids, tetraploids, and hexaploids in populations. However, we need to do some additional work to confirm this. Some of these estimates may not be as, based on microsatellite banding, may not be as accurate as flow cytometry. So we'd like to do some flow cytometry on this to confirm those ploidy variation. But if there is indeed ploidy variation, then there may be some issues with reproductive success because individuals of different ploidies often are not cross-compatible. So some additional crossing studies between individuals that look like they're different ploidies may be necessary to determine cross-compatibility and reproductive success. But that could be one of the reasons why this taxon is relatively rare, is because there could be some sort of limitation to reproduction. Okay, the next question was related to genetic structure. So first, we looked at clonality, and we found that of the 260 individuals we sampled, only 224 genetically unique wild individuals are actually remaining. And these individuals formed 3 genetic groups that were spread across 4 different sites, with sites being defined based on being separated by a mile. So 2 of those sites are unprotected, so 2 of the 4, so half, and those 2 sites contain a large proportion of the genetic diversity of a species, and they are not currently publicly protected. So I think those could be an important target for protection if that's kind of an option going forward. Another issue is that one of the genetic clusters is really imperiled because there's less than 10 individuals remaining in the wild that are from that genetic cluster. So those are the 2 smallest populations. And those 2 populations are in serious need of intensive management. So as I mentioned at the beginning, longleaf pine savanna is really needs to be intensively managed with fire and with clearing of kind of the mid-story vegetation in order to kind of maintain that open longleaf pine savanna structure. And if if that vegetation structure is not maintained, it will cause declines in the flocks, and that's certainly what's happened in these two smaller populations. They just have not been managed for flocks, and I think this is a really important situation where these populations need to be managed before they are lost completely. There's just a few individuals hanging on, and if they are lost, then we will lose a significant portion of the genetic diversity of the species. Another question was related to ex situ reintroductions and whether they encompass the genetic diversity found in wild populations. And another thing we found that was actually interesting is that they contain unique alleles um, that we think may have been lost from the wild populations. So these reintroductions are really valuable for conserving the genetic diversity that has actually been lost from wild populations. However, they don't necessarily encompass all of the genetic diversity present in the wild. And I think we essentially just need to do some more work to try to make sure that we're conserving all of the extant genetic diversity in ex situ collections so that it's not lost because the species does seem to be declining. Um, so we need to increase the genetic representation and genetic diversity in those ex situ collections or those reintroductions. Also, I think because the species is declining, and if we lose any of these populations, we could lose a really unique subset of the genetic diversity remaining in the species. We really need to do some conservation seed banking to make sure that we have a backup in the seed bank of the genetic diversity present in the wild populations. Okay, in terms of the conclusions related to the recovery of the species and whether we've met the recovery criteria. So I'll just remind you that the recovery criteria was that there should be 15 populations um, with 600 individuals in each population distributed across the three counties where the species is endemic. And I think I don't need to really say this, but the current status is that we have no populations that meet that criteria. And in fact, there's only 224 wild individuals in the species remaining. So we haven't even met the criteria for a single population, let alone for 15. Um, so unfortunately, there's really a lot that needs to be done to get to the point where we could even consider downlisting this species. So, urgent conservation, I think, is needed in this situation. So, for example, I think, a good priority would be to protect the two privately owned populations that contain a significant portion of the genetic diversity of the species, management of the two smaller populations to prevent their loss. Um, the next thing is whether we could maybe recover some wild populations in historical regions from the seed bank. So for example, if we go to a site where the species has historically occurred and we implement management, maybe we could get recruitment of the species from the seed bank. And even if we don't, then they would be great targets for reintroducing the species. So I think management in historic locations would be a good option. And then finally, I think the main take-home message is we need to conserve habitat for the species. We need to restore it and manage it so that it has an open understory so the species can survive. And then we need to do additional reintroductions to get to the point where we may even think about delisting this species because we're nowhere close. And in fact, it seems like it's going in the opposite direction. So hopefully, in conclusion, I've hoped I've shown how conservation genetics can provide some important insights into the limits and status, particularly for Texas trailing phlox. And I also just want to highlight how urgent action is needed to prevent the extinction of this unique taxon. Okay, so with that, um, I'd like to thank my collaborators on the project, George Eskevich and Bergen Basuner. I'd like to thank the Texas Parks and Wildlife Department for funding for the project. I'd like to thank Anna Strong for her help with the project, as well as Amber Beerb from the U.S. Fish and Wildlife Service, Sean Benedict from the Nature Conservancy's Sandy Land Sanctuary, who is almost single-handedly keeping this species alive, and then Anita Tiller from Missouri Botanical Garden for sending us samples, and then Andrew Bennett, Reagan Bounds, and Curtis Jackson for assistance with field work. All right, so with that, thank you, and I'd be happy to take questions.